Hello everyone, let's get started. Today we're going to be talking about eukaryotic cells. The eukaryotes, if you recall, have several key differences from prokaryotic organisms. We said that eukaryotes can be either single-celled or multicelled. They can be microscopic or macroscopic. Very different from the bacteria and the archaea. Some other key differences when we compare eukaryotes to prokaryotes are these. Eukaryotic cells contain organelles, including the nucleus, which houses the DNA. Remember, an organelle is simply a compartment within the cell that is surrounded by a membrane. And the organelles provide functions for the cell so that all of the work that a cell has to do every day can be compartmentalized using these individual organelles. Eukaryotic cells also contain a true cytoskeleton. The cytoskeleton is almost like a scaffolding inside the cell, and we sometimes forget it's there because the cytoskeleton doesn't show up with most of the stains that we use to stain eukaryotic cells in the lab. You have to stain very specifically for the cytoskeleton in order to see it. The cytoskeleton is actually made out of a variety of protein, and the proteins are organized into fibrous structures, linear structures, that help build this scaffolding system. The cytoskeleton has three primary functions that it performs for a living cell. Number one, it maintains the cell's shape. We've said that animal cells differ from plant cells, for example, in that animal cells don't have a cell wall around them. We often draw them as being circular or spherical to show that the only thing that surrounds that cell is a soft, fluid membrane. Plant cells, of course, are different. Plants have a cellulose cell wall around them, giving them a little bit more rigidity, a little bit more of a firmer, more structured shape. Well, the cytoskeleton is helping to keep both of those types of cells in their shapes. So maintaining cell shape is the first of the three functions that the cytoskeleton performs in a eukaryotic cell. The second thing it does is move molecules around within the cell. Now this may not be something you've thought of very much, but a lot of the materials that are inside a eukaryotic cell actually move around quite a bit. The cytoplasm, for example, doesn't just sit there like water in a stagnant pond. Instead, it actually circulates around the cell in a process called cytoplasmic streaming. Several of the organelles inside a eukaryotic cell are also on the move, as are lots of different types of protein and other dissolved structures. The cytoskeleton helps that movement occur. The last role that the cytoskeleton plays in the eukaryotic cell is physically moving the cell within its environment. We've talked about the kinds of structures that you see on the outside of bacterial cells and archaeal cells. Things like the fimbria and the pili and flagella. Well, eukaryotic cells also have external appendages that are just a little bit different in their structure. The eukaryotic organisms that are capable of moving through their environment have things like cilia on them, or flagella, similar to the flagella you see in bacteria and archaea. And it turns out that the cilia and the flagella that can help cells move are made from components of the cytoskeleton. Even cells like amoeba that move through their environment by extending bits of their membrane out into what we call pseudopods and lamellopods and philopods. These extensions of membrane help those cells crawl through their environment. The reason that those cells are able to extend their membrane that way is because of the cytoskeleton. So again, 3 functions for the cytoskeleton, generally speaking: maintaining the shape of eukaryotic cells, moving molecules within the cell, and helping to move the cell through its environment. Now, if we look at the genetic material in a eukaryotic cell, we also see some important differences between the eukaryotes and the prokaryotes. In a eukaryotic organism, the DNA is found in multiple pieces, multiple linear chromosomes. And recall that in a prokaryote, we typically see a single circular chromosome. Remember too that in a eukaryote, the DNA molecule, the double helix of DNA, gets wrapped around a particular type of protein called a histone protein. And once the DNA is all wrapped around histone proteins, it's very carefully stacked and folded into a structure that we call chromatin. Chromatin then resides in the organelle called the nucleus. We talked extensively about how prokaryotic cells have some extra chromosomal DNA. If you recall, those structures are circular and we call them plasmids. It is possible to find plasmids in a eukaryotic cell. Typically, you find them in the most primitive types of eukaryotes, especially the single-celled microbial eukaryotes. But even among those organisms, plasmids are rare. We also find extra chromosomal DNA in 2 of the organelles. Remember, we can find DNA in mitochondria, and we can find DNA in chloroplasts. And we believe that that DNA is there because those organelles were once free-living cells and became part of another cell through that endosymbiosis theory that we discussed. Lastly, it's important for us to remember that the DNA molecule in a eukaryotic cell is larger and more complex than what we find in a prokaryotic cell. So if we look at the double-stranded molecule of DNA, there are many more base pairs in that molecule in a eukaryotic cell. And if we look at those base pairs, there are many more genes in a eukaryotic cell. If you recall, we said a bacterial cell like E. coli typically has somewhere in the area of 4,000 base pairs in it. And in comparison, a human cell, for example, has approximately 3 billion base pairs in it. Remember, human beings, we don't even know how many genes are in human DNA yet. We have an estimate. We think it's approximately 30,000 genes, but we certainly haven't located and extensively studied all those genes yet. And we certainly don't know all of the different proteins that those genes code for yet. That work is ongoing. We talked about the characteristics that all cells share. We said there were 4 of them. And if you recall, one of those characteristics is these little cellular machines that we call ribosomes. We said both prokaryotic cells and eukaryotic cells contain ribosomes. Remember, ribosomes are not organelles. And the reason they're not organelles is because they don't have membrane around them. We find ribosomes in all cells, but the prokaryotic ribosome and the eukaryotic ribosome are just a little bit different from each other. They have some differences in their structure. So over on the right-hand side of this slide, you can see a drawing of a ribosome. Recall that ribosomes have two subunits. They have a large subunit and a small subunit. Now, in this drawing, you can see that the bulk of this molecule is this sort of a beige tan color, and then there are these red strings throughout both the large subunit and the small subunit. The artist is trying to show us that in both of the subunits, there is protein. That's what's in the beige, and there's also ribosomal RNA. That's what's in the red. So both subunits contain protein and rRNA. The difference between a prokaryotic ribosome and a eukaryotic ribosome is the exact kind of protein and the exact nature of the rRNA found in the different subunits. So this drawing represents a eukaryotic ribosome. So the artist has drawn the large subunit for us here and the small subunit here. And notice that each one of the subunits is labeled according to its size. So the large subunit is 60S and the small one is 40S. The S stands for Svedberg units. Those are the units of measure that we use when we describe ribosomes. And unfortunately, Svedberg units are not additive, because when we put the 60S and the 40S subunits together, we get a ribosome that has a total of 80S in it, not 100. So they're not additive. When we look at a prokaryotic ribosome, these Svedberg units are going to be different. Instead of a 60 and a 40, we're going to see a 50 and a 30. And the total ribosome combined in a prokaryote has 70 Svedberg units in it instead of 80. Again, the ribosome does the same job in a eukaryotic cell and a prokaryotic cell. It's there to perform the function of translation and to build the proteins that cells need every hour of every day. The difference between the prokaryotic and the eukaryotic ribosome is the combination of protein and the specific type, the specific nature of the rRNA that's combined with that protein in the ribosome. On this slide, on the right-hand side, we can see a depiction of several types of eukaryotic cells. Now, you've seen this drawing before. This was a drawing that you saw in the Introduction to the Microbes lecture when we were discussing the incredible diversity of size and shape of eukaryotic cells. Remember, we said the size and the shape of a eukaryotic cell is very closely related to the job it performs, to its function, in other words. So it makes sense that a neuron has these very strange sort of spiky appendages coming off of its membrane. These are called dendrites, because the function of this cell is to receive information from its environment and pass that information along to the brain and spinal cord. It needs these projections to gather all of that sensory information and pass it along. Remember, we said it makes sense that muscle cells, or what we call muscle fibers, are elongated like this because their job is to contract and relax. If you look at a generic eukaryotic cell, like the one depicted on the left, you can start to think about the individual organelles. It doesn't matter if we're talking about a neuron or a muscle fiber or a red blood cell or any other kind of eukaryotic cell. Because it's a eukaryotic cell, it has organelles in it. So the next thing we need to do is start thinking about these organelles, what they look like, how they're structured, and what their job is. If you look at this diagram, the artist has drawn an animal cell. Again, we typically draw animal cells as spherical. The artist has cut away a piece of the membrane so that we can peek inside and see these organelles. And the reason I like this slide is because, or this drawing on this slide, is because the artist has not only included all of these different organelles, but has drawn them in relatively reasonable size, comparing one to the other, and shown us that there's an incredible variety of organelles inside of eukaryotic cells. On this slide, we have a depiction up here of a plant cell, again cut in half so we can peek inside at the organelles. Remember, plants and animals, at least the land plants and animals, are eukaryotic. And so plants too have this huge variety of organelles inside of them. They also happen to have one or two organelles that animals don't have, most particularly this large vacuole here. But the idea is still the same. Plant cells, animal cells, all of the eukaryotic organisms on planet Earth contain organelles, and those organelles are there to do a particular function for the cell. I put this image down at the bottom here. This is an image taken of a section of plant tissue. If you had to guess, you could guess it was a plant because the cells are quite rectangular, not nearly as sort of spherical and amorphous as animal cells tend to be. The other thing you can clearly see in the image are these chloroplasts inside the cell. All of these small green circles are chloroplasts. So this bit of tissue comes from a green plant that is photosynthetic and is able to harvest the energy in sunlight to make its own carbohydrate. The reason that you see those organelles so clearly under the microscope is because they are full of pigment. They're full of chlorophyll, which looks green to our eyes. You'll notice that you don't readily see the other organelles in here. The other organelles have not been stained, and they don't contain this very handy pigment that would help us see them. So when we look at something like a plant under the microscope, typically the chloroplast is the only organelle that we see, unless we've done some special differential staining to allow us to see individual organelles. The other thing I want you to note from this image, which was taken under a light microscope, look at the size of the cells. This is one cell right here. And here's another one below it, right here. There's a third cell up here, which is partially out of your view. These are eukaryotic cells, and remember, eukaryotic cells are enormous when compared to prokaryotic cells. Plant cells are the most enormous of the eukaryotic cells.